Good morning, my dear students. Today we are discussing the continued part of induced breeding. A pair of breeder will be released into the breeding pond after injecting pituitary extract. The fish seed or egg is collected from the breeding pond. The spawn collecting net is called benchai jal or a funnel shaped structure which is consisting of closed mesh. In the first picture you can see the fish seed and the second picture you can see the benchai jal. Once the eggs get fertilized, they are kept into the hatching pits for hatching, and those are of two types. First one that is hatcheries and second one that is hatching hapas. So these hatcheries you can see in this picture, it is a small sized pond in which the fertilized eggs are transferred. After 12 to 15 hours, the fertilized eggs they get hatched. Hatching hapas. Hapas are rectangular trough-shaped tank which is made up of cloth and supported by bamboo poles fixed in the river. These eggs are aerated by continuous flow of water current. The hapas is made up of mosquito net cloth. Fish fries are transported to nursery pond by keeping them into fixed volume of water for a definite period, and then they are transported in open vessels. So here there are different ponds which works differently. So first pond, the name itself has its meaning, nursery pond, where exactly the spawn they get nursed or reared up to fry stage, and the pond is known as nursery pond. In the first picture you can see the fry stage, and beside that you can see the nursery pond, which is a shallow larger production pond where the water it is up to a depth of 2 to 3 meters. A layer of lime is spread over the bottom of the pond so that it removes the acidity of the soil and facilitates desirable geochemical cycles and kill unwanted soil organisms. The quality parameters such as temperature, oxygen content, pH, turbidity, hardness, alkalinity and plankton growth should be checked for their optimum level before stocking the fish. Manuring. Soon after liming, that is after 15 days, the manuring has to be done in order to develop the fish food organisms like phytoplankton and also zooplankton. Organic manuring it is also recommended. It is about 5,000 to 7,000 kg per hectare. Next pond, it is rearing pond where fries are reared up to fingerling stage or known as rearing pond. The first picture which shows the fingerling stage and second picture which shows about rearing pond. So rearing pond should be nearer to the nursery pond, and also the next pond what we are going to discuss, that is the stocking pond, so that they are easily transported. The fries they are grown up to fingerling stage of about 110 to 15 centimeter size for a period of 3 months. So fingerlings are reared in longer and narrower ponds to provide them long distance for swimming. So you can make out the difference between nursery and the rearing pond. The depth of the pond it is about 6 feet containing food material, and after attaining 20 centimeter length they are transferred to the next pond, that what we call it a stocking pond, where the fingerlings are transported from rearing to the stocking pond in a container of 1000-liter capacity, and while transferring they are made inactive by applying various sedative, and then they are transferred to the stocking pond. So the next pond where exactly the fish are stocked, that is a stocking pond, and this pond have to be cleaned of weeds and predator fish. Proper manuring should be done to increase the production of zooplankton because the rate will be high. Cow dung is provided at the rate of 20 to 25,000 kg per hectare per year. Chemical fertilizers like superphosphate, ammonium nitrate and sulfate are used at 1000 to 1500 kg per hectare per year. The amount of fertilizers used is totally dependent on the fertility of the soil, number of fishes, types of fishes being kept in the stocking ponds. The powder rice, paddy, oil cakes, mustard, groundnut are used as an artificial food. So once the fishes they have attained a length and a weight, they get harvested. So this harvesting can be done by capturing the fishes from the water. So well grown fishes are taken out for marketing, and the smaller one again they are released into these stocking ponds for their growth. So this is about starting from culturing of Indian major carps, moving with that is induced breeding and rearing of the fishes and eggs, that is fry stage, fingerling stage, and the fishes of maximum length and weight. So this will be included in culturing of Indian major carps. So like that, the next part, it is the culture of freshwater prawn, that is Macrobacterium rosenbergi. So here, apart from fishes, so aquaculture, it is not only culturing of fishes, as you know that. So it also includes aquatic crustaceans also for the economic importance. And the prawn fishery in India is next to USA in the world. And 150 species of freshwater prawns are found distributed all over the world. Among them, 40 species which belong to the genus Macrobacterium are found distributed in rivers, reservoirs, estuaries of India. Macrobacterium species like Rosenbergi, Rude, Mirabilla and Lamere, all these very important species, and Macrobacterium rosenbergi, which is having a faster growth rate and high tolerance to wide range of temperature. Because of that, they have very important value in culturing. So types of prawn fisheries, that is shallow water prawn fishery, estuaries and brackish water or saline lake prawn fisheries, freshwater prawn fishery, and last one, that is marine water prawn fishery. So characteristics of rosenbergi, so these are big sized prawn found distributed from lower to upper reaches of the river where there is a tidal influence, found in low salinity areas of estuaries. So this species prefers shallow and muddy environment and grows up to a size of 320 mm length and weighing about 200 gram. And you can see the difference between the male and the female. Mature males are not only larger than females but also possess second pair of walking legs. Juveniles are about 2 to 9 cm and can be identified by 1 to 8 horizontal lines on the carapace, which is a characteristic of arthropoda. Freshwater prawns undergoes the process of periodical molting, which increases the size of the animal. Next, coming to the life cycle. So the breeding occurs throughout the year, but the peak breeding, it is about March to May and October to November. During breeding, a male with hardened carapace, they mates with the freshly molted female. The male deposits spermatophores on the underside of the female thorax in between the walking legs. The female then extrudes eggs which pass through the spermatophores. The female carries the fertilized eggs with her until they hatch, and the female lay about 10,000 to 15,000 eggs up to 5 times per year. From these eggs hatches zoea, the first larval stage of all crustaceans. They go through several larval stages before metamorphosizing into post-larvae. This metamorphosis usually takes place about 32 to 35 days after hatching. These post-larvae then again they migrate back into the fresh water. So in this picture you can see the life cycle of Microbacterium rosenbergi with the brackish water as well as in the fresh water. So, collection of seed prawn. The post larvae or smaller juvenilines of rosenbergi are collected by nature with the help of fine meshed cloth nets. The seed prawn or eggs produced by collecting buried females, nothing but which are loaded with the eggs. Prawns with orange colored egg mass should be selected for the production of seed prawn. From this dark or grey colored females they are collected. The number of eggs in the buried female may vary according to the size of the animal. Production of larvae from eggs. So here first 3 buried females they are treated with 10 to 20 ppm formalin solution for about 20 minutes. Again they are introduced into a rectangular flat bottom tank which is of size 5 into 2 into 1 meter that should be filled with the brackish water. So these larval tanks are gentle slope at the bottom for easy draining of water. They are provided with the outlet at one side of the tank. The outer end of the drain tube it is fixed with a fine mesh cloth to avoid the escape of hatched egg at the time of changing water. The egg hatches out in about 4 to 5 days during early hours. After 3 to 4 days of hatching, at least 50% of water should be carefully drained off from the larval tank. And this larval tank should be provided with oxygen rich fresh water. Along with the pH should be checked. It is about 7 to 8.5, and also they are provided with different food, that is chopped earthworms, fish eggs, anople and moina larvae. After 10 days, these larvae start to swim upside down, and after 25 to 28 days of hatching, they become post larvae and swim normally. You can see in this picture that is the post larvae of rosenbergi. The post larvae are caught carefully and from the larval tank with the help of hand nets, and they are stocked. Here these post larvae they are reared in a concrete holding tank with fresh water at the rate of 5,000 per meter square, and after that once they start growing, that is after 1 month, the population can be made down by 2,000 per meter square. In holding tanks, the post larvae are reared for 1 to 4 weeks at the size of 1 to 2 gram, and there also they are supplied with the artificial food. So next one, that is transportation of seed prawn. So the freshwater prawns are packed in baskets layer of soft plants for transportation to nearby areas. They can live for several hours. If the journey is less than 1 hour, seed prawn are transported in 100 liter tin with about 40 liter of slightly brackish water in each. To such carriers, 30,000 post larvae can be transported. So if the journey is long, oxygen packed in polythene containers which consisting of both fresh water and brackish water, in that you can transport only 100 to 200 post larvae in one oxygen-packed polythene bag. So, management of production pond. So, the ideal size of production prawn culture, it is about 0.2 to 1 hectare. Water for these ponds may be from nearby wells or irrigation canals. Inlets are provided with screen or fine meshed nylon cloth to avoid the entry of predators. The quicklime may be applied at 1000 kg per hectare at a pH of 7 to 8.5 to increase the growth, and also to better fertility they are going to use the cow dung at the rate of 200 kg per hectare along with chemical fertilizers like superphosphate, that is 25 kg per hectare, may be applied every month during the culture period. Scrap nets or palm leaves may be installed as they form hideouts at various places in the production ponds. These hideouts also harbor organisms which in turn serves as a good food source to the growing prawns. The wild Ipomoea may also grow in small batches not exceeding 10% of total area, which serves as shelters for prawn. So once they have attained, so the next process it was stocking. For stocking in production ponds, 4-week-old juvenils, that is about 3 to 5 centimeters, are ideal. Before stocking them, these juveniles should be acclimatized in water of culture ponds for some time. A stock density of about 25,000 to 30,000 seed prawn per hectare, it is invariable adapted. So the culture operation may be either batch culture or continuous culture. Batch culture, that means the juveniles are grown only for 4 to 5 months and are harvested at one time, 2 crops in a year. So next, that is continuous culture. The name itself has its meaning. Juveniles are stocked once in every year and the marketable sized prawns are first harvested after the culture period of 4 to 5 months. The smaller individuals are released back in the pond. The culture is continued for another period of 3 months. So this completes the culture of freshwater prawn with an example Macrobacterium rosenbergi. Thank you.